Thank you very much, Edu. And I would invite Ed for the plenary talk, which we are very much looking forward, because we invited you. I already told him that he's among the Renaissance men who is studying basically everything about human behavior. So in case you are actually interested in, well, any aspect of human behavior, he's here to talk with you, to discuss. And we are looking forward to his talk. And now probably someone will need to help me. It's this one, right? Yeah. Yes. Got it. So this is actually working.
And then where's which button? Is it
it's only this one.
The forward button and the back button. Beautiful, thank you.
Okay.
Well, thank you very much for inviting me. I'm delighted to be here. And today I'm going to basically just describe or explain evolutionary psychology, at least the way that I perceive it. Many of the points I make will probably be familiar to most of you, but I hope at least I might provide a few novel insights for some of you. And my own personal goal or journey was to try and understand how do we understand human nature and human psychology and our emotions, our thoughts, as in a unified framework of all of nature. In other words, can we have a unified science, not just physics and chemistry on one hand and psychology and behavior on another, but can we all somehow view them as part of a single unified framework? And what I very quickly discovered is that, obviously I'm not the first person to think this, but this question goes back to the ancients. So we try and understand nature, the rocks, oceans, seas, clouds, hills, rivers, valleys, volcanoes, mountains, sand dunes, rainbows, mist, fire, water, drops, patterns. And to explain these sort of non-living kinds of things, scientists over literally thousands of years came up with abstractions, very powerful abstractions, things like objects that have a specific location in Cartesian coordinates, and that they have properties called mass and velocity and momentum and energy conservation and experimental methods. And these have all been spectacularly successful in explaining the natural world, or many aspects of the natural world. Here's Galileo's famous experiment, rolling balls down an inclined plane to develop formal theories of motion. Newton, conservation of momentum, conservation of energy, decomposing white light into its component parts. These are all the things that we learn in elementary school and high school. But it seems like understanding organisms like ourselves all have these properties. We have mass, we have location, we can have velocity, we have momentum, conservation of energy. But it seems like organisms have a lot of properties that are not easily explained with these kinds of abstractions. Those objects in the foreground there with the fins on them somehow seem qualitatively different from everything in the background. Can we somehow come up with principles that explain both of these? And it's not obvious how to do that. If we look at these objects in the foreground that we call penguins, they have all kinds of kind of strange properties. They're all nearly identical. They all have a very complex structure, but they all share the same structure. It seems very opposite from the random sort of patterns that we see in the background there. And one thing that, as I will demonstrate in a moment here, that has been recognized for, again, centuries or millennia, is that organisms have parts that somehow seem useful or purposeful, things like teeth and ears and fur. But where does purpose or meaning, where does this notion of usefulness, how does that enter into a naturalistic theory of the universe? We have, if we look at our skeleton, it's this extremely precisely articulated set of elements, of parts that seem organized in a very specific way to achieve some purpose, like grabbing things. And if we look throughout our bodies, we see that the same kind of structure characterizes everything, not only in us, but in all organisms that we've examined. We have all these parts that are useful. They're highly organized, and within a species, they're nearly identical from individual to individual to individual. So the musculatory system, the respiratory system, and they're organized in systems, the circulatory system, the lymphatic system. And then, taking on the wise comments that we just heard, organisms develop. Again, a very strange and unique property that we start with a single seed and this tiny little physical structure by itself, autonomously, will develop over a relatively short amount of time into this incredibly complex, sophisticated structure that can then create more of these little seeds, that then create more of these complex physical structures. What kinds of principles can explain all this? And of course, this is true not just with acorns and oak trees, but here we have little penguin chicks, and they are going to very reliably develop into that adult organism. We need some kind of organizing principles, but principles that are not unique to living things, but that are part of a framework to understand everything. And here, of course, we see our own teeth. This is an X-ray of a child's teeth. We have the baby teeth that have already erupted. We have the adult teeth coming in behind them. Again, a highly stereotyped, happens in all of us, very reliably. And these things, these teeth, seem useful, purposeful. How do we get this notion of purpose or function into science? So how do we do this? How can life and non-life be understood within a single explanatory framework? So, of course, I'm not the first to think of this, nor were evolutionary biologists. If we go back in time to Plato, Plato was worrying about this and thinking about this. And he noticed, this is in one of his early works, the Protagoras. He has his dialogues here, notices that different creatures have different properties, and there's a logic to it. Those that have strength are not swift. Those that are weak do have swiftness. Some are armed, some are unarmed, some are devised, have other means of protection. So he's beginning to put together a theory that organisms have these parts that are useful. But where do they come from? Well, for Plato, these parts come from the gods. The explanation is, in some sense, a teleological explanation. So writing later in the Timaeus, Plato proposes that there's a divine craftsman, a demiurge. And of course, there are strong echoes here with later Christianity. And this divine craftsman is rational, he's purposive, he's beneficent, and he imposes order on chaos to produce a vast array of good effects. So just to give you a quick quote here, the demiurge created the sun, moon, and stars to mark time, for instance, which itself came into being as an image of eternity. He created eyes. So Plato's explanation, why do eyes exist? Well, they exist because the demiurge created them to the end that we might behold the courses of intelligence in the heavens and apply them to the courses of our own intelligence, which are akin to them, that we might imitate the absolutely unerring courses of God and regulate our own vagaries. So the explanation for eyes are that the demiurge, who's this wise, beneficial, rational agent, created them for a specific purpose. This external agent is creating order out of chaos. So that's where the purposefulness and the utility comes from, at least according to Plato. However, Plato was not the only thinker grappling with these problems. There were the atomists, Democritus being a key example. And they rejected this teleological explanation. For them, the universe is explained by movements and interactions of a small number of particles, atoms, and they interact, and they had many ingenious arguments for how such a small number of particles and a limited number of interactions could explain everything we see in the world. And critically, there's no demiurge. Causation is only due to blind chance and necessity. This is very similar to our modern day physics. But it was very difficult in this framework to get to purpose and organization and function that we see in living beings. So Aristotle was grappling with both. He was grappling with the atomists and their arguments, but those arguments really didn't explain living things very well. And with Plato, who can explain living things as the product of a wise demiurge, but is bringing in this external being to kind of explain order, this external intelligence. So to explain some phenomena, Aristotle did invoke atomist arguments. Rain, for example, occurs of necessity and itself has necessary and chance effects. So why should not nature work? Not for the sake of something, nor because it is better so, but just as the sky rains, not in order to make corn grow. There's no purpose here, but of necessity. What is drawn up must cool, and what has been cooled must become water and descend. And the result of this is that corn grows. Similarly, if a man's crop is spoiled on the threshing floor, the rain did not fall for the sake of this, in order that the crop might be spoiled, but that result just followed. So for Aristotle, many things in nature are just chance and necessity, much as we might see in modern physics today, explaining much of the natural world. But Aristotle we needed an explanation for animals and the structure and purpose that we see. They cannot be explained only by blind chance or necessity. So teeth, for instance, are admirably constructed for their general office, the front ones being sharp so as to cut the food into bits, and the hinder ones broad and flat so as to grind it to a pulp. There's purpose here, there's function, there's organization. And because the teeth grow in this way in all humans, Aristotle goes on to argue, this pattern cannot be due to chance, contrary to the atomists. Instead, Aristotle concludes that there has to be what he called a final cause. Somehow immanent in the organism is some causation, some causal force that forces the organism to reliably develop teeth in every single member of that species. But there's no demiurge, there's no wise, beneficial creator. So Aristotle does have a teleological argument, but unlike Plato's, it's local, not global, and it's immanent in the organism, not external to it. And that's critical. And he wrote a whole book on this. So something is not just an aside, it was really a central focus of his scientific framework. Now Galen, famous Greek physician living in Rome, he was grappling with both Plato and Aristotle. So he accepted Plato's explanation that there is a divine craftsman. But unlike Plato, Galen also adopted many elements of Aristotelian thinking and understood that the structure of organisms are for the benefit of the organism itself. So unlike Plato, where our eyes are constructed by the demiurge so that we can observe the heavens, for Galen, who did many, many dissections, so he was very much an empiricist, and he did hundreds, if not thousands, of dissections of pigs and monkeys to try and understand the structure of those organisms, using empirical evidence on the structure of pigs and monkeys and assuming that they were very similar to humans, trying to understand how that structure benefited the animal itself. And he understood those interrelations of parts within the organism in materialistic terms, not teleological terms. And Galen's work was so influential, it really held sway, at least in Europe, for 1,400 years. So jumping over lots of history here, we get to William Harvey, who begins to understand all of these parts that Galen was grappling with in mechanical terms. So Harvey writing about the heart, the chief function of the heart is the transmission and pumping of the blood through the arteries to the extremities of the body. So Harvey is combining the increasingly sophisticated mechanical views of his age, but retaining the teleological aspects that these things are there for a specific purpose. Now jumping ahead again, I'm over a lot of history here, to William Paley. William Paley wrote a very influential book called Natural Theology. And he is increasingly developing this mechanical view of organisms. And so for Paley, there's still a divine creator. Of course, now it's the Christian God. But he's adding two things. Organisms are machines. They're not just like machines. Organisms are machines. They have all these parts. These parts have very specific relationships to each other. These parts benefit the organism, but they're also adapted to their environment. So the bodies of animals hold, in their constitution and properties, a close and important relation to natures altogether external to their own, to inanimate substances, and to the specific qualities of these. They hold a strict relation to the elements by which they are surrounded. Can it be doubted whether the wings of birds bear a relation to air and the fins of fish to water? They are instruments of motion, severally suited to the properties of the medium in which the motion is to be performed, which properties are different. Was not this difference contemplated when the instruments were differently constructed? So Paley is giving us a model, a conceptual model of organisms as machines that are adapted to their environments. And of course, here are birds clearly adapted to flying, and fish clearly adapted to swimming. And it was this model that Darwin read. As a young man, before he became a naturalist, he was assigned William Paley in class. And here's Darwin writing in his autobiography, crediting Paley. So just to quote here again, in order to pass the BA examination, and these are Darwin's words, it was also necessary to get up Paley's evidences of Christianity and his moral philosophy. The logic of this book, and as I may add, his natural theology, gave me as much delight as did Euclid. The careful study of these works, without attempting to learn any part by rote, was the only part of the academical course which, as I then felt and as I still believe, was of the least use to me in the education of my mind. I did not at that time trouble myself about Paley's premises, a God that did all this, and taking these on trust, I was charmed and convinced of the long line of argumentation. So in some sense, Paley has given Darwin the question that Darwin will then answer. Where do these machines that are adapted to their environments, how do we explain that, not in supernatural terms, but in naturalistic terms? And of course, many years later, here is Darwin writing in The Origin of Species. He gives us his theory of natural selection. We get organism design without a designer, without final causation. So just to quickly quote here, it is preposterous to attribute to mere external conditions the structure, for instance, of the woodpecker with his feet, tail, beak, and tongue so admirably adapted to catch insects under the bark of trees. In the case of mistletoe, which draws its nourishment from certain trees, which has seeds that must be transported by certain birds, and which has flowers with separate sexes, absolutely requiring the agency of certain insects to bring pollen from one flower to the other, it is equally preposterous to account for the structure of this parasite with its relations to several distinct organic beings by the effect of external conditions or of habit or of the volition of the plant itself. We need some other explanation, and of course the explanation that Darwin gives us is natural selection, heritable variation, and differential reproduction. We finally now have a naturalistic explanation for design without a designer, for organism structure, all of the stuff that philosophers and naturalists had been struggling with literally for millennia. We now have at least the beginnings of a naturalistic explanation. Now I'm jumping again here to Erwin Schrodinger. He's the famous physicist who gave us quantum mechanics. And he wrote a book that those of you who are interested in evolutionary approaches to organisms, I strongly recommend you read. It's called What is Life? It was written in 1944. It's actually a series of lectures that Schrodinger gave that were eventually published in a book. And this is before the discovery of the structure of DNA. So Schrodinger is trying to reason, how does the heritable variation and this heritable structure that is essential to the theory of natural selection, how can physics explain this? How is that consistent with what we know from physics? And so Schrodinger is starting with the problem: the unfolding of events in the life cycle of an organism exhibits an admirable regularity and orderliness, unrivaled by anything we meet in inanimate matter. How is this possible? And Schrodinger gives us two principles from physics how you can have order in what is essentially, if we look at it at the atomic level, a very highly chaotic and disordered universe. With thermodynamics and entropy and heat death, how do we get orderliness? And there's two ways, according to Schrodinger. One is statistical mechanics. This is order from disorder. So macroscopic structures, if you looked at them at the microscopic level, would be highly chaotic. But those highly chaotic motions of atoms would basically average out, and we can get stable physical structures like chairs and organisms. And so the organism, as a whole, must have a comparatively gross structure in order to enjoy the benefit of fairly accurate laws, both for its internal life and for its interplay with the external world. So we, as organisms, can have a stable structure, literally because we're so big. You could not build an organism from atoms. It would be too chaotic and random. You could only build organisms macroscopically, where we can take advantage of statistical mechanics. But that doesn't work for heritability, because from heritability we have a tiny fertilized egg that somehow contains everything it needs to develop from an acorn into an oak tree, or from a fertilized egg into a human infant. And so Schrodinger was struggling with this problem: what in physics allows that to happen? And he makes an argument in the book. I'll just give you the quick quote here. We can get order from order on the basis of quantum theory. And the basic thing is, I won't go over the argument here, but he shows how quantum mechanics can give us a stable molecule. And so he reasons that the substance, what he calls the aperiodic crystal, has to be a molecule that carries the hereditary information. Again, this is before the elucidation. This is before Watson and Crick. So just to quickly read here, it has often been asked how this tiny speck of material, the nucleus of a fertilized egg, could contain an elaborate code script involving all the future development of the organism. A well-ordered association of atoms endowed with sufficient resistivity to keep its order permanently, appears to be the only conceivable material structure that offers a variety of possible isomeric arrangements, sufficiently large to embody a complicated system of determinations within a small spatial boundary. Indeed, the number of atoms in such a structure need not be very large to produce an almost unlimited number of possible arrangements. So here Schrodinger is reasoning a priori, there's got to be a molecular code somehow inside the fertilized egg. Consequently, we may safely assert that there's no alternative to the molecular explanation of the hereditary substance. The physical aspect leaves no other possibility to account for itself and of its permanence. It is these chromosomes that contain some kind of code script, the entire pattern of the individual's future development and its functioning in the mature state. Every complete set of chromosomes contains the full code. So that code, of course, about a decade later, was now firmly established to be the molecule DNA. In humans, it contains 3 billion nucleotides. These are the molecular, or the atomic units, or the molecular units that Schrodinger was talking about. We all know them as the A's, the C's, the T's, and the G's. There's 4. So Schrodinger reasoned that there didn't need to be very many of these. In fact, there weren't. There were just 4. And there are a sequence of these, 3 billion of them. And we now know that about 1.5% of those specify proteins. That's about 20,000 genes, 1 gene per protein. So everything we're made of, every neuron in our brains, every cell in our bodies, are made of these proteins that are specified in this 1.5% of the DNA. We know another 10% of the DNA is non-coding. It somehow regulates, I'll talk a lot more about this, somehow regulates the expression of those proteins. We're still working that out. There's massive amounts of research on that right now. And it's also, it seems likely there's still debate on this, but maybe 85 to 90% of the genome might just be junk. It might really not contain have any real impact on the phenotype whatsoever. That's important too. And that's important for this. We have, I think, kind of in popular discourse, and also if I looked at my biological anthropology textbooks, there's two conceptual models of how we should think about DNA, and the most popular one, the most common one, the one that's taught in most textbooks, I think, is what I'm going to call the genetic difference model. I want us to reject that model, or not fully reject it. It's actually correct, but it's trivial, or compared to what I want to promote now, what I call the genetic program model, which is profoundly, this is the code script that Schrodinger is talking about. It has profound biological significance, and it's quite different from the genetic difference model, the one that unfortunately is more popular. So quickly, what do I mean by the genetic difference model? Well, this is the model where we look at genetic differences between individuals, and we correlate those genetic differences with differences in behavior, or differences in skin color, or differences in metabolic activity. And we do that between individuals, and we do it between populations. And this is kind of the basis for all of the genetic kits that many of us have used in the United States, 23andMe, Ancestry.com. I'm not sure what we have here in Brazil, but they tell us where we had some ancestors here, and we have some ancestors there, and we have maybe this risk factor for this disease, and this risk factor for that disease. And these are all based on genetic differences between two individuals. And again, it is true. There are genetic differences. Here they are. Here's a large study from the 1,000 Genomes Project Consortium. Each little tiny cross here, basically on the vertical axis, we have the number of differences between a specific genome and what's called the reference human genome. So there's a reference human genome, and then you sequence somebody else's genome, and you count up how many differences are there between this individual's genome and the reference human genome. And what you can see on the vertical axis, 3.8, 4, 4.2, those are millions of differences. And here we have a large number of individuals from the 1,000 Genome Consortium in different populations, Great Britain, Asia, African populations, Japan and so forth, Finland, Central Europe. And what you can see is that all of these various individuals have between 4 and 5 million genetic variants compared to the reference human genome. And I actually got a deal where I could get my whole genome, the whole genome sequence. I did that, and I got a file back, and the file had 5 million lines, and the 5 million lines are every point where my genome differs from the reference genome. And that sounds like a lot. But remember, there are 3 billion nucleotides in the entire genome. So 4 to 5 million variants is less than 1%. It's a tiny, tiny fraction of the entire genome. Most of our genome does not differ. And that's the part that is the most important. So let me just give you a diagram kind of showing where those genetic differences are. So in this, let's see if I can use my pointer here without screwing everything up. So these columns here, this is Africa, Europe, Southeast Asia, East Asia, and the Americas. These are the total number of, so here's the entire 3 billion nucleotides, 2.9, but let's say 3 billion nucleotides in the genome. Here is the percentage of the genome that differs across all of these different populations. So these are single nucleotide variants. So this whole thing is a zoom in on this little tiny fraction of the genome here. And this is the total number of differences, 91 million differences, not in a single genome, but across a large number of genomes. And then it's saying, let's organize those differences. Where do we find them? And so this whole chart is going to be from 0 to 100% of all of these 91 million differences. So these are variant counts. So 14 million of them, or almost 15 million here, 16%, are found in Africa, and they are rare variants. Now, what does that mean? That means that all these variants are almost only found in Africa. So you might think, hey, man, Africans have 15 million, almost 15 million variants that are not found in any other population. But they're rare. What does that mean? That means most Africans don't have those variants. These are basically new mutations that mostly that have very minor, probably negative phenotypic effects, if they have any phenotypic effects whatsoever, and they haven't been selected out. So the main message of this chart is that most genetic variants between individuals are rare, very rare, and they're population specific. And we can basically understand them as relatively recent mutations that probably have not yet been selected out of the genome. And only a tiny fraction of the total number of variants are common variants that we might find if we took any two individuals, that one of them would probably have these variants. There's only about 4% of the total variation in the human genome is actually common genetic variants. Most of it's rare. So yes, it's specific to Africa, it's specific to Southeast Asia or Europe, but even two individuals from Africa would not likely share those variants. Okay. So this is why I want to reject this model as being biologically significant. It's really not. Most of this genetic variation between individuals is noise. It's not the code script that determines the structure of the organism. That is the job of the genetic program model, and that's what I really want to emphasize here. Now I'm going to I have to do something a little tricky here to make this work. I had to do this to get the animation to stop. And let's see, I can now stop it. There we go. Okay. Many times when it comes to the word program, when people hear the word genetic program, they think of something that's very fixed and static. And so what I want to offer you is a little mathematical formula that is fixed. Let's see, where's my pointer here? This is the formula. This is the algorithm. This is the program that's running right here that we'll see in a second. Notice how simple this program is. This is called a logistic map. You can Google it. There's some really cool videos on this on YouTube. But it's super simple, and it's basically a model of population growth. X is population size. And we're basically saying that population size at time n plus 1 is equal to the population size at time n times a parameter, the growth rate, lambda here. So to get the population at n plus 1, you take the population at times n, multiply it by lambda, and you get n plus 1. But there's the second term here, and this is also a very simple term, that when the population starts off at 0, this just equals 1, and you just get the population growth rate here as we laid out. But as the population size gets close to 1, which is 100% of its maximum size, then this term is going to get smaller and smaller and smaller, and so you basically multiply the growth of the population by a term that represents that it's basically getting close to maximum, so that's going to decrease the growth rate as the population approaches its maximum. Now, what's interesting about this? On the vertical axis, we have the value of x. And then on the horizontal axis here, we have n1, 10, 15, 20, da-da-da-da-da. So this is the growth of the population over time. And what you're going to see is that this extremely simple program generates incredibly complex behaviors or outputs. So I'm going to play it here, and we'll watch it cycle through a couple times, if I can figure out what I'm doing. So it's already started a little bit, but maybe if I can, I'm afraid I'm going to screw something up. Let me try it from the beginning. Okay, I'm going to do this. I'm going to do that. Okay, now it's running. And what we're doing, oops, let me actually, I'll start it again. There's a little, what we're going to do is we're going to run it for different values of lambda, different values of the growth rate. So low values of the growth rate that are less than 1 are going to lead to the population to decline and go to stink. and values of lambda that are greater than 1 are going to lead to population growth. And what we're going to watch is an animation of what the population trajectory looks like for different values of lambda. So let's watch it. So as we can see here, lambda is less than 1, the population goes extinct. Now lambda is increasing, population is growing. But now watch what happens when lambda, this term here, gets over 3. We start to get incredibly chaotic, or variable behavior, not chaotic yet. But as soon as we get over here, it goes nuts. And so the lesson that I want to draw from this is not that this particular formula is of relevance to what I'm talking about, but just the general principle that very simple programs can generate enormously complex behavior. And what I'm going to kind of ask you to keep in mind is that this is a fixed program, but I want you to kind of think of lambda as an environmental input to the fixed program, and that'll become relevant in a minute here. Now let me get this going again here now. Oops, let's see, how do I start the slideshow? I'm clicking the wrong button, I think. Is that the button? Thank you. Okay. Oop, we got to get rid of that. It's gone, great. Okay. So now turning to development. We have a single fertilized egg. We're now talking about humans. And over the next 9 months, it's going to divide and divide and divide and divide and go from a single cell 9 months later to an infant with 2 trillion cells, all different. And then over the next 18 years into an adult of 30 trillion cells. And these cells are all different kinds of types of cells. So a single cell now becomes a multitude of cells, a huge multitude of 30 trillion different cells. Many of them are red blood cells, muscle cells, neurons, hepatocytes, bone marrow cells, lymphocytes, epidermal cells, and so forth and so on. So how does a molecule in this single fertilized egg determine this universal developmental sequence? And we're not going to go through in many details, but what I want to offer you for the genetic program model is that what's important about the genome are not differences between the genomes of individuals, but the interactions within the genome, within a universal human genome. So how does the genome interact with itself? Well, there's many ways, but the most important way is with a particular kind of protein or gene called a transcription factor. And these are genes that regulate other genes within the genome. And the basic way they work is that you have a gene that produces a protein called a transcription factor. But this transcription factor has a special, what's called DNA binding domain. It's basically a key. It's a special, highly specific, and each transcription factor has a different one of these different keys. But it has a very highly specific pattern that binds to very specific regions in the DNA. And by binding, by these transcription factors, which are produced by some genes, that then bind to specific regions of other genes, they regulate the activity of those other genes, either upregulating their expression or downregulating their expression. And these transcription factors can also have parts of them, they are proteins that bind to ligands, or ligands bind to them, that then alter their regulatory behavior. So the genome we should think of it as an incredibly complex machine in its own right, comprising parts of the genome that are interacting in extraordinarily complex ways with other parts of the genome to achieve very specific effects. And this is just a very simple example of one of these. These are called gene regulatory networks. And these are how we get all the different cell types during development. So here are some transcription factor genes that produce transcription factors that then bind to different genes that then produce more transcription factors You get a positive feedback loop here. And so then you get this positive feedback loop of producing proteins that will cause particular cells to develop into neurons, others to develop into muscle cells, others to develop into blood cells or lymphocytes or what have you. So there's a large number of networks defined within the genome that determine cell identity. Here's just a couple examples for interneurons and motor neurons. And we still don't understand these very well. These are still very poorly understood, but they are a focus of a massive amount of research right now. Now, to really understand these things, like we have to do with any physical system, you have to basically be able to manipulate the system. And we can't really do that ethically with human fetuses because you can't take a developing baby and say, oh, we're going to knock out this gene and see what it does. No. So we do it with model organisms. Sea urchins are very common. There's also mouse models. Here's a sea urchin developing from the early stages of 4 cells across time here. And this is by knocking out different genes. This is the circuitry, the gene regulatory network that was inferred that's guiding the development of the early sea urchin. And you can see how complex it is. And this is just part of the gene regulatory network or the genetic program that's guiding the development of the sea urchin. Same thing's going on in principle in humans. Now, a critical thing is that these gene networks can detect and respond to the environment. So here's just a very quick example. In our immune system, this is called a toll-like receptor. This is a protein that goes through the cell wall here, and it binds to different parts of bacteria, detecting infections from bacteria. Triggers a signaling cascade here, producing transcription factors that then alter the behavior of immune system cells to respond to a bacterial infection, for example. So a critical thing about these gene regulatory networks, they're not operating blind. They are like that logistic map where lambda we can think of as the environmental factor. That changing lambda, changing these environmental factors, alters the behavior of the gene regulatory network, often in profound ways. Now here's an example. These are water fleas. These 2 water fleas are genetic clones. They're genetically identical. But what you can see is that they're morphologically quite different. The one on the right here, or the one on the left here, has this very marked helmet that's missing on the one on the right, and this huge pointy tail, much different than what we see on the right here. So what's going on with these water fleas? Well, they live in a marine aquatic environment, and they are predated upon by fish. And building this helmet is very expensive. It takes a long time, a lot of resources. You don't want to build the helmet if there's no fish, if there's no predators around. But if there are predators, you've got to build that helmet early on. You can't just develop it at the drop of a hat. You can't say, oh, there's a predator, I need a helmet. You need to do that early on in development. So water fleas have a gene regulatory network that can detect chemical cues of predators. If those chemical cues are absent, no helmet develops. But if the chemical cues of the predator are present, the water flea develops this defensive helmet. Now you might ask, is there anything like this in humans? And I'm going to argue we have many examples in the room here. We have males and females. And males and females are basically created by the same genetic program, but that just gets altered due to a genetic switch. So here's sex determination in mammals, including humans. You get the initial development of the genital ridge here. If you happen to have, now we all think, hey, I've got a Y chromosome that has all the genes to code for male. If I don't have it, I develop into a female. That's actually not how it works. There's only a single gene on the Y chromosome that determines if you're going to be a male or not, and that's called the SRY. And if you have it, it triggers a gene regulatory network on chromosome 17. It basically binds to what's called SOX9 here and develops a whole regulatory cascade that leads to testicular development, And if you're lacking SRY, you then go down a different regulatory path and get ovarian development. But critically, all of this are on the autosomes. They're on the genes that are shared by both males and females. And this has been known for well over 100 years, that basically our genomes contain all of the code to create a male or a female. And it just depends on the presence or absence of this genetic switch, SRY, not on the other genes on the Y chromosome, of which there are actually very, very few. And I want to just offer this as possibly, we need this in our conceptual toolkit. We know about the water fleas, we know about sex determination in humans, but we might have other things like that detect environmental cues and trigger different developmental trajectories. So many of you probably know about fast and slow life histories. Could this be due to some environmental switch? Or maybe even a genetic switch? Possibly. We actually have evidence in our immune system that our immune systems develop differently if we're in very rural kinds of environments versus more urban environments, for example. So this is just an idea that we should keep in mind, that there might be different developmental pathways beyond just sex or beyond just the immune system that are sensitive to environmental cues early in development. Now, these gene regulatory networks guiding development, they're not just in us humans, of course, they're very conserved. Conserved means they don't really change genetically, even between mice and humans, or across all mammals. So these gene regulatory networks, we can learn, are very, very conserved. They don't change. And that means within humans, they're essentially identical. So our basic model is we have this whole genome. These are the green squares. There's a tiny amount of genetic differences. That's mostly noise. And then different kinds of inputs to these gene regulatory networks, SRY, genetic switches, epigenetic switches, environmental conditions, trigger the program to cause it to behave in different ways to develop a phenotype that is tuned to local environmental conditions. In the water flea case, are there predators or not? In the human case, who knows? We know maybe in our immune systems, are there lots of pathogens or not? We might need different phenotypes to deal with those. Okay, how do we bring this back to now psychology? Everything I've said applies to our entire phenotype, not just our psychology. How do we bring this to psychology? How do we have a machine model, like a pump of the heart? What's the analogy or the analogous sort of mechanism or machine for the brain? And of course, as we all know, it's the computer. So the computer is our model of the machine that is the brain, that is cognition, that is psychology. And the different parts of our brain are not levers and pumps and valves, but instead what we might think of as apps. Just as your smartphone is a single computer but has a lot of different apps to do all kinds of different things, you need different apps for taking pictures, for writing notes, for talking on the phone, for sending messages. The argument here is that we need different apps in our brain to deal with all the different information processing challenges that humans faced over the course of their evolution. So this is evolutionary psychology, that we have multiple gene regulatory networks that evolved by natural selection to generate tuned psychological adaptations that are tuned to our local environment, apps, algorithms, modules, pick your term. These solved important problems of survival and reproduction posed by, and for things to evolve by natural selection, it takes a long, long time. So it had to be problems that were recurring over lengthy periods of the ancestral environment. So what is this environment? Well, here we have a plot of fossils, human fossils, or fossils of human ancestors, I should say. And I'm kind of missing a little element here, but we'll make do. On the y-axis, so each dot here is a fossil. So Lucy is down in here somewhere. Well, we don't have Neanderthals in this particular plot, but they would be up in here somewhere. Homo erectus down in here. These are millions of years before the present. So this is today, 1 million years ago, 2 million years ago, 3 million years ago. And what we have on the vertical axis is the brain size of these fossils. And this is the normal the modern chimpanzee range is right about here. And the modern human range is right around here. And what we can see is about 3 million years ago in the Pliocene, our ancestors' brains were about the size of chimpanzees. And then over the next 2.5 million years, starting right about here, this is the beginning of the Pleistocene right here, and going over this 2.5 million year period, our brain size triples. And where is that happening? It's happening in Pleistocene Africa. So the major structures of human cognition are evolving in this environment in the Ice Age, in the Pleistocene, right here, as you can clearly see in this plot. So, can we know anything about that past? Well, yes, we can. So here's just some basic climatic data. On the x-axis, again, we have time. Here's today. On the vertical axis, we have global average temperature. And this solid horizontal line here is the average temperature today. So values above this are warmer, values below this are cooler. Here we have the time of the common ancestor of all primates. You can see the environment was very warm and wet, maybe plus 12 degrees back then. And then the climate undergoes a long period of cooling and drying over this long period. So here, about 6, 7, 8 million years ago, we have the common ancestor of human and chimps. And here's where we have those fossils I just showed you, starting right around here. And the first members of our genus Homo show up right about here, 2.5 million years ago. And this is when brain size begins to increase. And what you can see is that the climate is, we're entering the Ice Age. And not only is it cooling, but over this period when human brain size triples, it becomes extraordinarily variable. And many people, myself included, think this climate variability has a big part to do with that increase in brain size. And modern humans show up in Africa right around here, 200,000 to 300,000 years ago. And then we get the end of the Ice Age, and here we are today. Now that's just temperature. But how strange was this Pleistocene African environment? Well, it really wasn't that strange. Here's just an artist's depiction of all the animals and landscape that we would have seen in Ice Age Africa. And we can see it looks extraordinarily familiar to what we would see today in any kind of savanna environment. There's animals, there's plants, there's birds, there's large mammals, there's small ones, there's water, there's rivers and lakes and ponds, there's clouds, there's rain, there's hills, there's forests, there's grasslands. It's an environment that's really almost exactly what we see today. It's not that exotic. So modern environments are reasonable proxies for the Pleistocene environments. And the kinds of environmental features that were relevant to human survival today were relevant then. Physics, mass, gravity, light, sound, motion, fire, weather, temperature, rainfall, wind, geography, lakes, rivers, resource locations, infectious diseases, people, faces. There were infants, there were children, there were adults, there were males, there were females, there were kin, there were non-kin. There were groups, there were animals, there were arthropods, there were parasites, there were tetrapods, birds, mammals, predators, prey. And there were plants that had leaves and fruits and seeds and toxins. So all the kinds of things that are critical to survival and reproduction today have been critical to survival and reproduction for a long, long time. I keep going to the keyboard here. I don't need to do that. Got the button. Now, of course, many people would say, what about learning? We can learn about all these things. Yes, we can and we do. But learning is not simple. And this is just a classic model of Pavlovian conditioning. And I just throw it up here to show you. Learning is complicated. It's a complicated system to be able to learn useful information from the environment. That requires specialized psychological adaptations to do the learning. And the same goes for culture. We need specialized adaptations to learn culture, use culture, and transmit culture to others. A blank slate doesn't learn, a blank slate doesn't learn culture. There needs to be cognitive structure to do those things. So let me just give you some examples of how we can apply these principles to begin to explore new areas of evolved human psychology. This is some great work by a colleague of mine, Annie Wirtz. And the basic adaptive problem is that plants are important sources of food, but many are highly toxic and poisonous. So how do we deal with that problem? So in experiments that Annie has done, she wondered, how early in development do we begin to recognize that plants might be dangerous? So she works with young infants in this kind of paradigm, and it's a very simple thing. She takes these young infants and she puts in front of them different kinds of objects. So some of those objects are natural objects like rocks and seashells. Others are artifacts like trowels and spoons and keys and hairbrushes. Others are novel artifacts that the kids would never have seen before. Some are artificial plants, and some are real plants. And then she simply says, how long does it take for the infant to reach out and touch the object? And what you can see here, these are the different kinds of objects here. So here we have natural objects, familiar objects, novel objects, artificial plants, real plants. And then this is the delay of the infant to reach out and touch the object. And what you can see is they relatively quickly touch these things, but they take a lot longer to touch these things. In fact, they won't usually even touch them. They look to the adult to see if it's okay. So this is at least one piece of evidence that infants are more cautious around plants than other kinds of objects, and the explanation might be that they have an innate knowledge that plants can be dangerous, that could kill them. Okay, here's a second example, remembering locations of food. Where is the food? You've got to have specialized memory to remember where food is. So in this experiment, they took individuals into a farmer's market that had a number of different stalls with different foods, things like lettuce and cucumbers and cherries and oranges and avocados and honey. And they take them through the market. Each student, or each participant, I should say, goes a different path led by a researcher, and they taste a food at each stall. And then after that, they are taken into the middle of the market where there's a big dial, and they have to kind of point. They say, okay, where was the lettuce? Rotate the dial and show me where the lettuce was. Okay, tell me where the blueberries were. Rotate the dial and show me where the blueberries were. And so forth and so for all the foods that they've tasted. And then what we have here is the error, the pointing error. How far off are they? So 0 is no error. They exactly locate the direction of the food stall with the food. So this is the error. And then on this axis, on the x-axis, is how nutrient dense is the food. And what you can see is there's a very interesting pattern. People are pretty inaccurate with foods that have low caloric content, lettuce, cucumbers, zucchinis. But they're quite accurate with foods that have high caloric content, honey, almonds, olive oil. So this is at least some evidence that we might prioritize memorizing the locations of nutrient-dense foods over those that are much less, have far fewer nutrients. Okay, third example. Animals are also an important source of food, but they can be dangerous. So in this study, it basically does a study where you see if you can detect a change in a scene and how quickly you can detect it. So there's a mask, and then you see a scene for just a short amount of time, a quarter of a second, and then there's another mask, and then you see the same scene, but something has changed in the scene. And the question is, how long does it take people to detect what has changed in the scene? So using Photoshop, the researchers just changed a tiny little element of the scene. Well, what did they change? Here's some examples. And what they change is the presence or absence of an animal or a vehicle or a person or a bird. Here's a little bird here. Here's a person here. And they basically Photoshop, or a cup here, and they Photoshop these things out. So you're seeing these pictures flipping back and forth, and you have to say, okay, something has changed, what is it? And what they find is, and then how accurate are they? Sometimes they never detect it. And what they find is people are very quick and very accurate at detecting that what has been removed is either a person or an animal. And they're much less good or accurate at picking out even things like vehicles or buildings, like in this one. They took out this whole building, and people were very slow to detect that an entire building had disappeared. So the argument here is that we are specialized to detect people and animals when they appear on the scene because they are opportunities but also threats. Okay, final example, mate choice. This, of course, is what evolutionary psychology is notorious for, sex. Finding a mate, we don't just mate randomly. Nobody does. We don't just close our eyes and pick the first person of the opposite sex and have sex with them, or the first person, or the first object. We usually put a huge amount of time into picking mates. And there are some qualities that are universal, that there's no predicted sex difference. So this is a very large, and there are other properties where we do predict a sex difference. And this is a large study. It's actually a replication study across 45 countries for mate preferences. And what we have here, and each dot here is one country. And what we have here is the sex difference. So this would be, it's males minus females. So if the female scores are bigger, we're shifted over to the left. If males are bigger, we're shifted over to the right. And for the properties that are predicted to be important for both sexes, like health and kindness and intelligence, we see a little bit of a sex difference, but not much. This is where we have no predicted sex differences, and indeed the sex differences are quite small. Males and females are very, very similar in preferring mates that are healthy and intelligent and kind. But we do have some predicted sex differences. We predict that females are going to prioritize, so both sexes want resources, both sexes want fertility, but due to pregnancy, females are predicted to put a greater emphasis on resources and males on fertility. And this is what we see, good financial prospects. There's a female bias, a very clear female bias. So both sexes like this, females like it more. Physical attractiveness, which is thought to impact fertility, both sexes like this, males like it more. And then age choice, which is both a combination of fertility and resources. There's a very consistent that females prefer mates that are a few years older, not that much, but 2, 3, 4 years older than them, and males prefer mates that are a few years younger than them. The argument being that younger females have higher fertility and older males have more ability to produce resources. So we see, again, some at least evidence for human universal mate preferences here and sex differences in the predicted direction. But these sex differences tune to the local environment. So this is, again, the same data. Each dot here is one country, but the countries are now separated out on the x-axis here by the gender equality of the country. And we can see that in very gender unequal countries, the sex difference in age, or the sex difference in age preferences is quite large. But as gender equality increases, the sex difference decreases. So as the differences between the sexes decrease, the preferences for an age difference also decreases, suggesting at least some evidence that mate preferences are tuned to the local social environment in this case. Now, hopefully it doesn't need to be said, but I'll say it anyway. We've got to avoid the naturalistic fallacy. Just because nature is one way, just because human nature is this way or that way, doesn't mean human nature should be that way, that it's good to be that way. Aggression exists, jealousy exists, all kinds of bad things are probably part of human nature, but we are very justified in trying to reduce those. This is from Hume, and then from Moore, very similar thing. Just because something is natural doesn't mean it's good. Okay, to conclude. Organisms comprise parts that were designed or engineered by natural selection to promote survival and reproduction in the EEA, the environment of evolutionary adaptiveness. And the brain, so that's the organisms in general, and the brain is the same way, no difference. It has parts, psychological adaptations, that were designed or engineered to promote survival and reproduction in the EEA. And these parts are generated and tuned by universal gene regulatory networks that take environmental inputs. So remember, what is fixed in evolutionary psychology are these gene regulatory networks, not even the psychological mechanisms, and definitely not behaviors. Remember that logistic map. The formula is fixed, but the lambda can change, and that can dramatically change the behavior. So we became human in the African Pleistocene, we all did. It posed many adaptive challenges. And that gives us these universal gene regulatory networks that create the phenotypes that we all have today. Thank you very much. Have I left any time for questions?
Thank you very much for a wonderful talk. There is really lots of food for thought. I think that I will need to watch this talk again, like at least 3 times to digest everything. But are there any questions right now? Oh, and if you want to, you can also, the local people can ask in Portuguese if you want to. We can try to translate. You don't need to be afraid asking in English, if that would be a problem.
Ed, thanks for your presentation. Just a few minutes before your presentation, Ottoni talked about kind of consilience between different approaches to evolution of human behavior. Do you think that, for example, the perspective of evolutionary psychology of a reliable development and recognizing this difference between other approaches on the evolution of human behavior which not work with a reliable development, so acknowledging these differences can lead to this consilience, for example?
Yeah, that's a great question. First of all, I would say all these approaches are generating data. And I would say we're probably all wrong. Evolutionary psychology is probably wrong, ethology is but we're all right in a certain way. So that the data, the evidence generated by all these different approaches are going to have to be reconciled in some framework. I would also say evolutionary psychology grew out of ethology, and it grew out of a lot of these other traditions. So I would say some of that consilience is already happening here. And I guess the third thing I would say is empirical tests. We have to really, we kind of come up with these grand frameworks, but at least my own experience with my own projects is I get this great idea and then the data don't support my idea. So I'm very much, very strongly in support of robust empirical programs that are really focused on testing ideas. And in your studies, I encourage everyone, when you put together a study, don't just test your idea from evolutionary psychology or from ethology, but take different hypotheses from the different traditions into the same study and test them against each other. And that way is how I think we will achieve the consilience that Eduardo is looking for in our experimental approaches and our empirical approaches, that we don't just look at our own approach, but we take the hypotheses from the other approaches, put them in the same study, and pit them against each other head to head. And what I often find, at least in my own studies, is nobody's right, and we have to kind of come up with a new theory. So I would hope, yes, that we are all steadily marching toward a consilient framework that will probably look a little bit different from anything we see today, would be my guess, but we'll see. Thank you. Yeah.
That's a very positive view, which I would like to share. Other questions, comments, suggestions?
So I'm very new to all of this. Just about the sex thing, like 2 slides ago, something like that. Yes. Does it take in account like the patriarchy? Like men are like being told over and over, like, yes, like young girls to prefer, like the socialization interferes in the results? Or it's just like, oh no, it's just biology?
So in this study, each dot here is a different country. So there's 45 different countries here. And you're right, what we see here is in this chart, each country has a gender equality score, which would be, in some sense, a measure of the patriarchy. So highly patriarchal cultures are here on the left, where there's very a male-biased power. And then the countries here on the right are much more, this would be, much more gender equal countries. And what you can see in terms of the sex difference in age preferences is they decrease as gender equality increases. So this is the sex difference here. These are females up here who would prefer partners that are about 2.5 years older than average, and these are the males here who would prefer partners that are about 2.5 years younger than them. And what you can see as you get more gender equality, males are preferring partners that are closer to their own age, and females are too. So yes, there is an impact, you're absolutely right. But we still see a sex difference.
Yes.
So will it disappear completely? Who knows? But this is the idea that our psychological mechanisms may be tuned to the local social environment. As the social environment becomes more equal, mate preferences seem to be becoming more equal as well. You're absolutely right.
Thank you.
Yeah.
Thank you for your great talk. My questions come from ignorance. I'm not a biologist, I'm not a psychologist, I'm a linguist. I'm here by chance, so to say. But I have some questions, mainly basic questions. One is, which is the, let's say, ontological nature of this gene regulatory networks? It is something biological. This is the program built in the gene.
Yeah. So what this is.
The other one is, which is the difference between this approach in which you talk about tuning to certain conditions and epigenetics?
Epigenetics, is that what you said? Yeah. So the networks, that's a great question. Where do they come from? So we can think of our DNA as a linear string of 3 billion symbols, A, C, T, and G. And this linear sequence is divided into what we call genes. So each gene specifies the structure of a protein. And those proteins, some of those proteins, not all of them, but some of them are what we call transcription factors. And those proteins, those genes that code for transcription factors, the transcription factors have a special structure called the DNA binding domain, and it's like a key. And so it will only bind, so this gene produces a transcription factor, which is a protein. The protein has a special section of it that has a kind of a key-like structure that will only bind to certain areas on the DNA. And when it binds to those areas, it causes that gene to either produce protein or not produce protein. It kind of turns the gene on or off. And different transcription factors have different keys that fit different parts of the DNA. So the gene regulatory network is the set of these transcription factors that have specific binding domains that connect to different parts of the genome, which then connect to different parts of the genome, which can connect to different parts of the genome. So it's that pattern of interconnections determined by this kind of lock and key system that determine the networks. Does that make sense? Yeah. So that's where the gene regulatory networks come from, is this pattern of binding here. Now, in terms of epigenetics is a rather generic, general term, and so many biologists say, this is epigenetics right here. This is the genome these are proteins regulating itself. Another aspect of the epigenome would be that, and this is I didn't get into, but DNA, about 1.5% of it actually codes for protein. Some of those are transcription factors, but a lot of DNA gets transcribed into RNA, and you have to know what RNA is, but basically the chemical process goes from DNA to RNA to proteins. But you also can have DNA to RNA and not going to proteins. This is called non-coding DNA, and that RNA can also bind and regulate the expression of genes. So that makes we have another element that I didn't talk about here, the RNA elements, the non-coding RNA elements that also regulate the genome. And then there are some other things like methylation that can bind to some of those ATCs and Gs, and again, turn on or turn off genes. So yes, epigenetics is definitely part of this. It kind of depends on your definition of epigenetics, but right here. So we definitely have mechanisms by which things can influence the expression of genes, elements. We can have chemicals in the environment, as we saw with these guys. Just chemicals that are out in the environment can alter the behaviors of these gene regulatory networks. But there can also be products, other cellular products, proteins, RNA, other ligands produced by the body that can alter the behaviors of these gene regulatory networks, absolutely. That's critical to the whole concept. Did that answer the question? Sort of. We can talk more if you'd like later.
Now, it's kind of a continuation of this, what you just said. No? But for me now, after your explanation, made me think, even got a bit confused, some concepts you showed. Like, for example, what is the difference between genetic switches and epigenetic switches?
Yeah, I think that's a good question. So, and again, it really depends. Unfortunately, epigenetics is a term that's not very precise. And it really kind of means so genetic, everybody would agree transcription factors are produced by genes, they produce proteins, they regulate. So they may exclude transcription factors because those are genes regulating genes. But what about the RNA that is not, in some definitions of a gene, it's not producing any protein, it's getting transcribed, but it can also regulate DNA. And what about methyl groups that get imprinted, that attach to certain parts of the genome, that can also alter expression of genes? And then our genome is wrapped around, these are called nucleosomes, we're wrapped around these proteins, so there's a packing structure. And those protein packing structures are called histones. And to actually express DNA, you have to kind of open up the DNA, open up this package, and so you can get histone modifications that are modifications of the packing proteins that can also alter. So your question is actually a good one. How do we distinguish genetic switches from epigenetic switches? It really depends on which lab you're working in and how they want to define epigenetic. It's really kind of an unfortunately vague and ambiguous term. I saw something on Twitter recently that had I think it was a 4x4 grid of various epigenetic definitions. So there's 16 different definitions, and depending on which tradition you were in a different cell. So I kind of don't like the term epigenetic because it's just not very precisely defined. That's about as good as I can do to answer that question.
Você traduz? Eu vou falar em português, você traduz. Eu queria saber, na opinião dele, o quanto ele acha que nós, psicólogos evolucionistas, estamos conversando bem e fazendo pontes com as pessoas que fazem epigenética, porque eu acho que é muito gene centrado ainda, essa ponte que a gente tenta fazer.
She wants to know what you think about the relations we as researchers in evolutionary psychology.
I will try. So,
Okay.
we as evolutionary psychologists, in your view, in your opinion, how do you think if you are doing a great job in building bridges with, I don't know, epigenetic colleagues to talk, I don't know, because I think we are very much gene-centered. Even though we know that epigenetics are in the picture, but in my view, I think we must building much more bridges, talks, and understand much more to, I don't know, talk about this kind of thing, because I don't know, I consider myself much ignorant in this matter to know, I don't know. There are a lot of definitions in epigenetics, but I consider myself ignorant. I have to study much more. Do you think that we can build a strong build with this area, or we are in our infancy to build this?
So what we see right now is we've, and I'm not sure if I fully understand your question, so feel free to.
Sorry about that.
That's okay, no, I feel free to jump in and steer me on the right course if I start heading in the wrong direction here. But, you know, in the last couple of decades, we sequenced the entire human genome, and we've now developed technologies for sequencing the entire genome for a few $100s. So we now have tens of thousands, maybe even hundreds of thousands of complete genomes. And that has given us, we basically now know what genetic differences look like between people. And that really hasn't taught us very much about what it means to be human. And what you see if you just dip into the literature is that there's a massive amount of research to try and understand these gene regulatory networks, to understand gene expression. And they're developing what are high throughput techniques to look at cell by cell by cell, which genes are being expressed in which cells and when. And it's a vast amount of research, but it's extraordinarily complicated. We're very much in the very, very early stages of putting this together. So even with, you know, these guys, sea urchins, we can only do the first few hours, and even then we're not 100% sure what's going on. And this is a system where we can actually, it's ethical to manipulate and knock out genes. So yes, we're in very, very early days of understanding this. So even though I think there's good reason to believe this will be the foundation, and maybe for very limited problems, it might help us. I think the real value for me of evolutionary psychology is this picture, which is if you looked at psychology prior to evolutionary psychology, it really wasn't asking what I would consider to be fundamental. How did humans survive and reproduce? What kinds of mechanisms do we need to solve all these problems? So there was all kinds of things on cognitive biases and sort of various ill-formed questions about social relationships, but it really wasn't getting at fundamental problems of survival and reproduction in the kinds of environments in which we evolved, which are often very similar to the problems that other animals were facing. And so for me, the value is really thinking about this and how this might explain universal psychological structures as we saw in like these examples, like this, I love this plant research, and infants just somehow innately knowing that plants might be dangerous. So I think for me the value of evolutionary psychology is kind of turning the ship around to begin asking the right questions. It doesn't necessarily give us the answers, but it's helping us ask the right questions, I think.
Thank you so much.
Yeah.
I have one more question. I'm sorry to keep bothering you.
Go ahead. No, that's fun for me.
Must be
So
times.
please.
Now, one thing that I didn't understand, and is still in my mind, that you said at the beginning of your presentation, is about the like 95% of DNA is noise. What do you mean by that?
Great question.
It's junk.
Yeah, great question. So, sorry. There's a technical term called conserved. And what conserved is, we know that mutations happen randomly. And we have a mathematical model called the neutral model, which says if mutations are happening randomly and there's no positive selection, there's no negative selection, then the statistical patterns of genetic variation should match what's called the neutral model. So it's basically a model of noise. And what you can do is you can look at, we now have all the genetic data from large numbers of humans. You can look piece by piece across the genome, and you can look at the patterns of genetic variation, and you can say, are those patterns consistent with what's called genetic drift, or just random mutations that are happening that are not being positively selected or not being negatively selected? They're just happening and the kind of random processes that, and if that's true, that means that those areas of the genome really are probably not having any influence on the phenotype because they're not influencing survival and reproduction, therefore there's no selection, therefore the patterns look totally random. And so that's what we would call not conserved. And when we look at the genome section by section by section, it looks like about 85% of it, or maybe 90% of it, is evolving neutrally under drift, that mutations are accumulating randomly, and they're not undergoing positive selection, they're not undergoing negative selection, which means they probably just don't have any impact on the phenotype whatsoever. And only about 10% of the genome looks to be conserved, which means that if a mutation happens in that 10%, it usually will be eliminated by natural selection. So what we see is 10% of the genome is surprisingly missing genetic noise. There's no variation. You and I would be identical. So if there's a gene where you and I are identical across the all the, let's say, 500 amino acids in the gene, it's probably because that if any one of those changed, we might not even get through development, or we would not survive and reproduce. That's called conserved. So about 10% of the genome looks like those are areas where when mutations happen, they're quickly eliminated by natural selection, which means they have an important impact on the phenotype. So when we look across the entire genome, it looks like, and there's still a debate about this, so I don't want to put an asterisk by this, but it looks like about 10% of the DNA is critical, and whenever mutations happen in that part of the DNA, they're quickly eliminated by selection. But the other roughly 90% of the DNA, when mutations happen, they just happen and they stick around, and they don't have much impact on the phenotype whatsoever. Or, as this diagram showed, they may have an impact, but it's very minor. So most of these genetic variants are rare, which means these mutations have happened. They haven't been so severe that they've prevented the fetus from developing or the individuals from surviving and reproducing. But they're either what we call neutral or very mildly deleterious. Did that answer your question? Sort of? Okay. So basically the conserved parts are the important parts that really influence the phenotype, and the non-conserved parts really don't influence the phenotype, and it really don't have a big impact on survival and reproduction.
So thank you very much for the discussion, and thank you, Ed, for accepting this invitation, for giving us this wonderful talk and discussing with us in the meantime. And we brought you a small present so that you can do your own caipirinha at home, which always helps, right? We need to increase the well-being of our speakers. So thank you again.